All right, cool. So good afternoon, everyone. Welcome to another exciting deep dive presentation. So today what I'm going to be talking about is B cells and what they do. So hold on. Okay, there we go. So what we're going to talk about today is a brief review over innate and adaptive immune system. We're going to look over B cell maturation and activation and differentiation. And then we're going to talk about antibodies and terminology. So to begin, we have the innate immune system. And this is involved, this works daily in your body to remove different types of foreign particles or pathogens that come into your body. So a lot of times you don't even notice, but your immune system is always working, and unless it's really high in amount of foreign particle titers, will you ever really realize that you are under a disease? And so most of the time, your body will be able to clear this with these different leukocytes. Um, so in terms of allergies, it usually basophils, eosinophils, and mast cells that will activate against allergies. And then for neutrophils, this is usually first responders for bacteria, fungi, and macrophage usually come a few days later, and they tend to clean up any cell debris and can act as antigen-presenting cells. Then you have dendritic cells, which mainly act as antigen-presenting cells. They also phagocytose different pathogens, and they're like they're the bridge between the innate immune system and the adaptive immune system. And so I think I showed this image before, and basically when an infection occurs through a opening, through a little scratch or injury, the cells in that area will begin to release cytokines. And it's these cytokines that can signal your neutrophils and your monocytes to go into the site of infection and start, uptaking these different pathogens, hopefully being able to clear them out before they spread too far. But if the pathogen is able to grow where your innate immune system is not able to handle it, this is where we activate the adaptive immune system with the dendritic cells that are able to uptake and present it to these specialized cells. And so what makes adaptive immune system different from your innate immune system is that these cells are able to distinguish different pathogens and remember them. So they're able to develop a memory against these pathogens. And so a lot of times the basis of vaccination is to target these cells. And so you have two different pathways that can be activated. You have the humoral immunity and the cell-mediated immunity. The cell-mediated immunity is what we talked about last time. This is T cells. Usually helper T cells get activated, and they go and activate cytotoxic T cells, or they will recruit macrophage and natural killer cells to go ahead and kill any cells that you have that might be infected by causing pores and forcing those cells to apoptose. And so today we're going to focus more on the humoral immunity. And again, this does involve helper T cells, but mostly this is the B cells that are able to produce antibodies. And this is usually for extracellular microbes that are going around the body, releasing different toxins and growing in your body. So I guess what I want to talk about is, I like to give credit to those who discovered these different immune cells. And so last time we talked about Dr. Jack Miller, who discovered the thymus is able to create T cells. And so before, everyone thought the thymus was a useless organ, and he showed that the thymus is very important in producing T cells. He also later discovered that there was communication between T and B cells. But before he even could figure that communication, we had to talk about Dr. Max D. Cooper. And Dr. Max D. Cooper was the scientist that discovered B cells. And so I'm going to talk a little bit about his life. So below you can see that there's a review where it's autobiography about his life, and it was a very interesting autobiography. So if you find time, you should read it. I think it's very inspirational. He was a pediatrician who worked at a hospital for sick children, and his move or his change in area to academia was based on the knowledge that we did not have on the immune system. Um, there was a lot we didn't know and how to cure diseases, and so he really wanted to investigate and look into that more. So he actually applied to be a postdoc in Robert Good's group, and Dr. Good is credited to being one of the founders of modern immunology. And so Dr. Cooper basically restarted his life at the age of 30 to become a postdoc. And he never had any lab experience. So in his autobiography, he mentions that he felt underqualified and less than a lot of the other postdocs that had lab experience in that lab. So a lot of his time was basically catching up and trying to understand what was known about the immune system at that time. And so what is very interesting is at that time we believed that there was only one lymphocyte that was responsible for all the different differentiations that we can get now, which are T cells, natural killer cells, B cells. We thought there was only one cell lineage for them. And what Dr. Cooper, as he gained more knowledge and got more experience doing working with his lab mates, he decided to do an experiment where he took newborn chicken and he irradiated them to remove any pre-existing lymphocytes that were developed while they were in the embryonic state. And then what he did was he removed the thymus or the bursa of Fabricius or both in chickens. And so what this did was when he removed just the thymus after irradiation and looked at the lymphocyte population, he found that the chickens still had antibodies and plasma cells. And then when he irradiated the chicken and took out the bursa of Fabricius, he found that there was still a population of lymphocytes that could perform cell-mediated immunity. And so when he removed both, there was no population of these lymphocytes. And this is how he discovered that the bursa of Fabricius was another population of lymphocytes, which he called B cells. And then the T cells were from the thymus. So he was the one who kind of named this scheme. One thing that people would question him at this time was, yeah, great that you found this organ that produces B cells, but what's the equivalent in mammals? And so the rest of his time when he went into becoming a professor was kind of finding the equivalent of the bursa of Fabricius in mice. And so what he found was in mice, the equivalents was bone marrow and the fetal liver. And now as a professor at Emory University, he's looking at understanding antibody class switching in B cells. And he's actually trying to find the origin of where the immune system kind of started. So he's looking at lamprey, which are a lineage of jawless fish. And he's trying to he's found that there's a older version of the immune system in these lamprey, and he's trying to now use them for maybe diagnosing current treatment. So that's kind of what he's working on now. And so like I mentioned, for primary B-cell development, this occurs in the bone marrow or fetal liver. And what we're going to really focus on is the bone marrow, because after you're born and your liver grows, most of your B cells are going to be developing in the bone marrow, where they come from hematopoietic stem cells. And only 5% of these will actually go on and be able to complete the primary development to actually go into the circulation as mature naive B cells. And so after the hematopoietic stem cells grow, they become common lymphoid progenitors, and then those can either become natural killer cells, T cells, or B cells. And so the way that works is the hematopoietic stem cells that have a low level of PU1s become common lymphoid progenitors, because hematopoietic stem cells can become all leukocytes. So they can become neutrophils, natural killer cells, dendritic cells, but if they have a low level P1, they become a common lymphoid progenitor. These CLP can bind to stromal cells in the bone marrow, and if they receive interleukin 7 and B cell activating factors, this will then start the B cell development. If this does not occur, then it either becomes a T cell or a natural killer cell. And then afterwards, these B cells will undergo BDJ recombination. And I'll talk about this more later on, but this is what is required for the B cell receptor to be unique from other B cells. And it undergoes two different selections in the bone marrow. It has a positive selection and a negative selection. And so the B cells that undergo positive selections have to receive signals and be able to transduce these signals. If it does, it moves to the next step. And then the negative selection is when the B cell receptor is able to not activate to self-antigens. If it activates to self-antigens, it either apoptose or it will undergo further BDJ recombination to have a B cell receptor that doesn't activate to self-antigens. And so once it makes it to the different developments in the body or in the bone marrow, it'll then move into either a secondary lymphoid organ, such as the lymph node or the spleen. And so for the secondary B cell development, we talked about the bone marrow part, and I guess I should show my laser pointer. We talked about this primary B cell development. And so the secondary B cell development occurs in the spleen and the lymph node. And so basically what a T1 B cell is a transitional 1 B cell. This is when the B cell moves from the bone marrow and migrates to the extrafollicular area of the spleen or the lymph node. And in this area, it's going to encounter a lot of blood circulation. So it will be able to interact with more blood proteins. And so again, here it's being tested to whether or not it activates against self-antigens. If it does, then it undergoes apoptosis or becomes an anergic B cell, which is labeled as T3 B cells. If it's able to not activate, then it moves over to a transitional 2 B cell. And these B cells are when they migrate from the extrafollicular area or the outside areas of the lymph nodes and spleens, and they go more internal into the center of the lymph nodes and spleens. And here they can start becoming activated to pathogens and foreign particles that enter the body. And then they can either become marginal zone B cells or follicular B cells. And this is basically where these are two different subsets of B cells and the location in the lymph node that they may stay in. And so then the way that these different subset of B cells that are now considered mature, naive B cells because they're not activated by a pathogen yet, can either activate in two ways. One, the B cell can receive the pathogen first through its B cell receptor and then phagocytose it. And they will display this on their MHC2 molecule to present it to a CD4 helper T cell. And this will then activate the B cell to proliferate into memory and plasma cells. Another way that this can occur is if a dendritic cell takes up a pathogen, activates a helper T cell, and this activated helper T cell will then find a B cell that it can activate to differentiate into plasma cells or memory B cells. And so a lot of times we need to be able to track these B cells, and in vaccination especially, what we do is understand whether or not we're activating our B cells and developing plasma cells. And so one of the CD markers we look at is CD45R and /B220, and this is expressed on all B cells. And so to differentiate them, you can then add other protein markers that are on the surface of different B cells to distinguish them from the overall population. And for activated B cells that we look at CD9, we can look at CD25 or CD30, because CD19 is actually present in most B cells as well. For plasma cells, we can differentiate them with CD78 and CD138. And then for memory B cells, we can look at CD20, CD40. We can even look if they have the IgG or IgA on their B cell receptor. And then for regulated B cells, we can look at CD1, CD5, or CD21 along with CD45R and B220. So now I'm going to move towards antibodies because this is the main role of all these different B cells is to produce these antibodies. And in each step, the B cells will produce a small amount of these antibodies and secrete them out. But most of the antibodies produced are produced by plasma cells. And so what we want to look at here is the B cell has its fragment antibody binding fragment here. And this is what binds to the antigen. And it binds to the variable light chain and variable heavy chain. And so each antigen can bind up to 2 different foreign particles on its binding site. And then what we also have is the fragment of crystallization, or the fragment of crystallization. And this is this area here. And this area is very important in being able to bind to Fc receptors. And so the Fc receptors, there's a bunch of different Fc receptors on different immune cells. You have Fc gamma receptor 1, Fc gamma receptor 2, Fc epsilon receptor 1. And so the gamma means that it binds to IgG1. Fc epsilon binds to IgE. And so this can basically cascade different methods of activation or what it wants that immune cell to do. So for like IgG1, when it binds to the Fc gamma receptor 1, this induces macrophage and neutrophils to uptake the antibody pathogen into phagocytose it and basically neutralize it or destroy it. And we talked about BDJ recombination. And so what BDJ is both the light chain and heavy chain have this. For light chains, it only has V and J. For heavy chains, it has V, D, and J. And the way it works is you have 40 different variable regions. You have 27 diversity, which is for D, and you have 5 different joining regions. And your DNA will then excise different regions so that at the end of it, you have only 1 V region, 1 D region, and 1 J region. And then you have your constant region. And so this basically allows your B cell variable region, this allows your variable region to be different amongst different B cells because in this case, we're showing V2J2 for the variable light region. And so this will express a region that has a specific binding to a specific pathogen. But if it's B4J5, it's going to have a different binding to a different pathogen. And so that's what makes every B cell receptor unique. And so there's like about 10 to 11 different combinations that can occur, making each B cell receptor unique to another. But one of the problems with this large combination is that some of these combinations can lead to B cell receptors that can bind to your self-antigen. And so sometimes it has to undergo BDJ recombination again to remove self-antigen binding B cell receptors. So I'm going to go further, and now I'm talking about class switching, which I forgot to mention here. Is class switching is dependent on the Fc fragment. And so you have different antibodies or different type of antibodies. You have micro, gamma, alpha, delta, and epsilon. And this, again, like I said, it can bind to different Fc receptors and cause different activation for those immune cells. And the way you change the antibody class that you want is through a process called, with an enzyme called AID. And so what basically occurs is here you see the VDJ, and then you have the constant region. And in the constant region, you see micro, delta, gamma, or IgG3, IgG1, IgG2b. And so what happens is the AID enzyme will excise the sequence that it doesn't want to express. So initially, you'll see that it expresses IgM and IgD. But after it excises this region and removes it from the DNA, now you'll see that it will express your VDJ, and then it'll start expressing IgG2B. And this is how your B cell will class switch. And so your antibodies have three different ways of interacting with foreign particles. This is through neutralization, and this is when it basically just binds to the pathogen, not allowing it to enter into another cell, and basically excretes it out of your blood system. Then you have opsonization, and these antibodies will bind to pathogens, and then the antibody will bind to an Fc receptor to tell the immune cell to phagocytose it. And your last method is a complement activation. And this is where the antibodies bind to the bacteria, and through a cascade of events, it basically causes pores on your pathogen, forcing it to apoptose. And so you have, like I said, you have different antibodies. And so now you can see how these look. So IgM is the first antibody that's produced by a B cell. And usually when it's secreted from the B cell, it's in a pentamer form. So it has 10 different areas where pathogens can bind to. When it's on the B cell membrane, it's only a monomer. Then this is basically responsible for early stages of immunity. IgD, they're still trying to understand what IgD does, but they feel like this is part of an older system, or it's just there. It doesn't really have a function, but they're still investigating what it actually does, or what its role is. Then you have IgG, which is present. It's one of the most abundant immunoglobulins. It's present in the blood, and it's actually able to transfer from mother to child. And this is called passive immunity. Then you have IgA, which is a dimer antibody, and this is present in all of the mucous membrane lining. So it's present in like the gastrointestinal tract, in your bladder. So this is IgA. And then you have IgE. And so initially IgE is thought to be a antibody against parasitic worms. But I guess since we don't deal with that as much, a lot of allergic reactions are caused by IgE. So I'm going to give you some more terminology for antibodies. And so antibodies are considered a lock and key mechanism. So like I said, every antibody can bind to a specific antigen that is able to fit into its antigen binding site. Then we have monoclonal antibodies and polyclonal antibodies. And what the difference is monoclonal antibodies can only bind to one side of an antigen. And these are produced by one B cell that has differentiated. And so this is very unique and very specific and selected to that antigen. Now, polyclonal antibodies are produced by several different B cells, and it can bind to different areas. And so what makes polyclonal antibodies, they're easier to produce, and these can have a lot of cross-reactivity between different antigens. And so cross-reactivity just means that it can either react to different epitopes in the same pathogen, or it can cross-react between different pathogens with similar structures. And then we have affinity versus avidity. Affinity means how well one antigen binding site can bind to an antibody or epitope region, and how strong this binding is. Where avidity is the overall interaction of the antigen to the antibody. And so, for example, IgG has a strong affinity to epitopes, and IgM has strong avidity. And that's why IgM is a good B cell, is a good immunoglobulin to produce, because it can bind to multiple places, increasing the strength of binding. So now we're going to talk about the how plasma cells and hold on how plasma cells and memory B cells are produced. And so we talked about the B cell being activated by dendritic cells, then talking to the T follicular helper cells. And then once they activate, they actually form a germinal center, or they proliferate. And then these proliferation goes to a germinal center. And the importance of a germinal center is that somatic hypermutation and class switching recombination occurs. So this is where B cells can improve their binding to a pathogen. And again, the way they do this is they make a slight change in the DNA sequence by changing one cytosine to a uracil. And when it does this, since uracil is not present and can't be read, there's a enzyme that's called, give me a second for this, it's called uracil DNA glycosylase. And what it does is it switches that uracil into another base pair and completely changes the sequence, causing a change in the B cell receptors variable chains. And this can then increase the binding to that antigen, or it might make it worse. Or sometimes it can also cause it to react to self-antigen. So it's very important that in this dark zone where the germinal center is occurring with these mutation, that the B cells migrate back to the light zone and interact with T regulatory cells and T helper cells. Because if there is self-antigens, the T regulatory cells will cause these B cells to apoptose. If the B cell improves its binding to the antibody, the T cells will give it positive reinforcement to continue to proliferate and go back to the germinal center to undergo more mutations to see if it can improve it more. And in some cases, those improved B cells will then become memory B cells and long-lived plasma cells. So there's different types of plasma cells. There's plasmablasts, and plasmablasts are different from plasma cells in two ways. Plasmablasts are able to proliferate, and plasmablasts still have a B cell receptor present on its membrane. Plasmablasts can produce antibodies at a higher amount than B cells, but they are relatively short-lived. And usually before entering the germinal center, plasmablasts are formed to help fight the pathogen that has invaded. The short-lived plasma cell will come from the germinal center. These have stronger binding antibodies, but again, short-lived plasma cells and long-lived plasma cells do not replicate and do not have B cell receptors on its membrane. Short-lived plasma cells tend to remain in the lymph node and the spleen, and they only tend to live several days to weeks. Some of these short-lived plasma cells that migrate to the bone marrow will become long-lived plasma cells that can survive for years. And this is one of the newest cells that we found within, I think, the last decade or two, and what we're trying to aim for in vaccinations. So I thought that was very interesting. And two new cells subsets that we have found recently too are T-independent B cells and regulatory B cells. And so T-independent B cells just means that they do not need T cells to be activated. And the way that these B cells activate are typically to bacteria and viruses. The antigen or pathogen has to have multiple epitopes that are repeated so that it can bind to several B cell receptors and toll-like receptors at the same time. When binding to both, it is able to be phagocytosed and it can activate the B cell to proliferate. Now, one thing about T-independent B cells is that they cannot produce plasma cells or memory B cells. And so this is why they think that these some bacteria don't create memory is because they're activating independent B cells. But there's other literature that contradict this and show that independent B cells can form germinal centers and they can form memory B cells. but this is a new area, so there's still a lot to understand. And then we have regulatory B cells. And what regulatory B cells do are the same as regulatory T cells. They downregulate inflammation by excreting interleukin 10 and TGF beta 1. And when the interleukin 10 will downregulate MHC2 expression, and interleukin 10 will also downregulate IFN gamma and TNF alpha, which is very important for Th1 response and Th17. So regulatory B cells basically lower the pro-inflammatory response, and they also are, at least in literature, are thought to be the reason of activation of regulatory T cells. So regulatory B cells will remain in the lymph node and spleen, basically regulating that area, and they will help activate regulatory T cells to regulate areas where the infection is occurring. And so that's what those two different regulatory cells do. So now I'm going to move on to something interesting. This is a paper I read about dosages. And so this paper talked about the different concentrations of a pathogen. And what they used was flagellin, which is an H antigen in bacteria. And they injected mice with different dosages of this H antigen. And what they see is an inverse response. What they showed was if you inject a low amount, you tend to have a stronger cell-mediated immunity. But once you start increasing the dosage, you see that the cell-mediated immunity response go down and the humoral response go up. And so what they reason is your body prefers a humoral response over a cell-mediated immunity response. And this is because in a cell-mediated immunity response, your body tends to attack infected cells. So the more virus or bacteria in your body, if your body was to go through a cell-mediated route, you would have more cell death, and this can cause necrotic tissue, and this is what kills yourself. So to prevent your own immune system from killing you, your immune system tends to switch to a humoral response when there's higher amount of bacteria or virus titers. And I think that makes sense. And so now I'm going to talk about another paper, which is a very interesting paper. I think the remainder of my talk is going to just be about this paper. What they focused on was looking at a subunit vaccine or subunit protein for HIV. And in this paper, they wanted to change the way we looked at dosaging. And most of the time, what we do for dosaging is one shot, followed by a booster shot several weeks or several days later. And so in this experiment, they looked at a constant injection, a exponential decrease followed by a booster shot, and an exponential increase followed by a booster shot. In all cases, the antigen delivered was the same concentration at the end. And so what they found was when they compared this to the prime shot and booster shot, you see that the exponential increase and the constant delivery had a higher amount of antibodies at day 14. And you can even look at when they track the antibodies over time. You can see by day 14, there's a higher amount of antibodies created. But after the booster shots, you see that everything tends to increase to a similar amount, except for the exponential increase showing slightly higher antibodies.
Hey, Michael.
Yeah.
How did they deliver the exponential increase and decrease? Is it like a sustained release method, or how did they do that?
So every day they would inject a larger amount subcutaneously.
Okay, so it was like multiple deliveries?
Yeah, exactly.
Okay. And are those like IV or intrapreneurial injections?
These are subcutaneous injections.
So, okay, that's what you said. Sorry about that.
No,
okay,
no,
cool.
it's okay.
Thank you.
It's totally fine. Of course. And so then they wanted to test something else. They're like, okay, we did it every day for a week, and now let's do it every other day to do a 2-week interval. And so what they did was, again, injected it every day, and then had a booster shot, and then they did it every other day with the booster shot, and then they have their controls. Again, when they compare it to the controls, you see that there's a higher amount of antibodies for the 2-week interval versus the 1-week interval. And you can look at their antibodies over time, and you can see that the 1-week and 2-week interval are having a higher production of antibodies. And they continued this. Oh, sorry. They continued it forward with this, and they then did another experiment with mice where they did it with a 2-week interval since they saw a higher amount of antibodies. And then instead of doing a booster shot with just one injection, they decided to do that exponential increase for the booster shot instead. And again, they're showing that as you exponentially increase it, you get higher antibodies. And they reason that when you have an actual infection, the virus will start to, proliferate. And so you don't see a decrease, or usually for vaccinations now, we do a 1 shot followed by a smaller booster shot. And that doesn't make sense because your body's going to have, when a virus is there, it's going to continuously increase. So far, this study makes sense, and that's the way they reason it, that this is why this works. Then they looked at the lymph node to see the amount of antigens in the lymph node at 24 hours and 72 hours after the injection. And again, what they showed was the antigen at 24 hours were about the same when they looked at the lymph node. But after 72 hours, you see that more antigen is available in the lymph node compared to the prime and booster shot, or prime shot. They did some computational studies, and this was a little bit hard to understand. But what they did is they used an equation of free antigen binding to IgM, forming an immune complex. This immune complex would then bind to B cells, which is needed to activate the B cells to produce plasma cells. These plasma cells would then develop antibodies, and then again, we look at antigens that bind to IgG to form different immunocomplexes that can further activate the germinal center. And so for C, what they're looking at is the free antigen. And so when they inject this subcutaneously, computationally, they show that the free antigen disappears right away in all cases. But the immune complexes formed are higher for the exponential increase. And so the more immune complexes there are, the more activation of B cells will be obtained. They also showed that computationally more plasma cells are developed, and IgM and IgG are produced at higher amounts for these for the exponential increase injections. And finally, and I think most importantly, the total antigen available in the lymph node based on these computational modeling shows that there's more total antigen in the lymph nodes for the exponential increase. So they're saying that when they model this computationally, they see the same results that they're showing in vivo. And so what they did next is they wanted to look at the lymph node of the 2-week interval versus the single bolus shot. And so what we're seeing here in C is the germinal center B cells. And so you can see that the naive has less than the bolus shot, and the bolus shot has less than the 2-week interval. And so by looking at the PNA and GL7, that's the positive cells. Those are germinal center B cells. And you can see that there's a lot higher for 2-week interval. Then they looked at plasmablast. And for plasmablast, they had the cell has to express CD138, but it doesn't express B220. And so again, they showed that the 2-week interval had a higher amount of plasmablast compared to the bolus injection. And this is just the number of cells that they have on the right of that of the flow cytometry data they got. And then E is looking at activated B cells. So this is CD86 positive and MHC2 positive. And so what they found here was the bolus at day 1 had a higher amount of activated B cells And if you look on the right, they actually looked at day 7 and day 13, and they showed that the activated B cells go down, but the 2-week interval has a higher amount of activated B cells by day 13. Then they looked at the lymph node to show the amount of B cells, and which are labeled with blue, and then the amount of antigens, and they showed that the antigens are in higher amount in the lymph node for the 2-week interval compared to the bolus shot. So again, they moved forward, and what they wanted to do was kind of do they implanted a mini pump, and this mini pump would deliver the antigen over a long period of time. So instead of doing daily injections, they did this pump instead, and they did a 1-week interval and a 2-week interval. And what they looked at again was T helper cells in the germinal center, and what they showed here again is that the 14-day interval had a higher amount of T helper cells compared to the 7-day pump and the bolus shot. And then they also looked at the overall number of just T helper cells in the lymph node, and again they showed that the mini pumps had higher amounts than the bolus shot. And then when they looked at the germinal center B cells, again, in each case, they're showing a higher amount in the pumps compared to the bolus injection. They also looked at the IgG titers, and they showed that the pump, and this is the 14-day pump, has a 15-fold increase compared to the bolus injection by week 12. And they looked at something called the B3 specific IgG antibodies. And these B3 specific antibody, or this B3 specific region, is a region of denaturation in the subunit vaccine of HIV that can sometimes occur when injected in the body. And what they showed was that the bolus injected mice have a higher amount of this V3 denatured protein, and the pump actually protected them from getting this antibodies, which is actually a good thing. So I just want to end it up with just saying that B cells, from what I've read, are great to understand, so we can start treating autoimmune diseases, helping prevent allergies, which is what we already do. Start looking into cures for type 1 diabetes, and improve vaccinations overall. There's still a lot we don't understand about B cells, and we're still finding new subset of B cells and trying to understand where they come from. For example, we don't understand where regulatory B cells are developed and made. Some people believe that they come from the marginal zone B cells, but we're still not sure. So a lot of studies are trying to understand how they develop, or what causes the B cell regulatory cells to be created. What we're trying to understand for vaccination is how do we attain long-lived plasma cells, and then also understanding how to combat the aging immune system. And so there's this theory that your immune system can only develop a certain amount of memory B cells and memory T cells. And so as you combat more pathogens, older memory B cells and T cells apoptose to make room for these new memory B cells that your immune system is currently fighting against. And so if that is true, how do we prevent that, or how do we improve our memory without losing old memory to pathogens? So I hope you guys enjoyed this talk, and if you guys have any questions, I'm more than happy to answer them. Thank you.
Thank you, Michael, for that very informative talk. I had a question. Where are germinal centers formed in the body? Sorry, that's kind of a silly question, but I was just kind of curious. Are they like in the blood or in the tissue?
That's not a silly question. I actually did not know really about that until I started reading. Well, when I started getting into this area. But germinal centers are formed in the lymph nodes and spleen.
Okay.
So they can be formed in both areas.
Okay. That makes sense. And then I had a question, since we've been talking about B cell activation, in your list of cytokines, I didn't see CD80 or CD86.
Yeah.
Do you have any information about those two in regards to B cell activation?
Well, what is thought is that for activated B cells, they do upregulate CD80 and CD86. So if you want to look at activated B cells, I would suggest looking at those
Okay,
markers
so they're good
alongside.
for activation.
Yeah, and I would look at it alongside MHC2.
Yeah, yeah, we talked about that. Okay, that's a good point. All right, thank you, Michael. Very nice talk.
So I see that Jeremiah asks, is there a difference in the FC region for neutralizing versus opsonization antibodies? Um, I think that, so I'm not entirely sure. I know that each FC receptor will cause, like I know for IgG, it tends to be more for opsonization, but I'm not sure what FC region or what class would be better for neutralization. Maybe it's still an IG, but maybe a subset of the IG that is more prone to neutralization. But that's something I'm not entirely sure about. Because I know that IgGs are more prevalent in the blood. And so I think whether it's neutralization or opsonization, I think those are both uh, should be they should both be IgG, but maybe different subsets, if that answers the question, I hope. But I do need to look into that more.
Just to, I guess,
Yes.
ask my question from the like specific way. So in like all of these models on the slides with antibodies on the outside of the plasma cell, I, I guess I'm kind of viewing that as the plasma cells are like scaffolds for the antibodies. Is that the wrong way about looking at it? And that the
Wait, wait.
cells are made dependent on the disease, or they can develop different antibodies based on what, I guess, disease is present. They can multiple antibodies or multiple antibodies for different diseases on one cell, or is that
So,
confusing?
yeah, so the B cell receptor will only bind to, actually it. So we I think we like to think that there's only one B cell that will bind to that pathogen, but I don't think that's the case. I think you have several B cells that might have close or similar B cell receptors that can bind to different epitopes of a pathogen. And so I think these B cells will then move into the germinal center and basically compete against each other to find the B cell receptor that binds, or that B cell that binds best to that pathogen. And that B cell will proliferate. And when that B cell proliferates, it can become a plasma cell. And that plasma cell can then release antibodies specific to that pathogen.
Okay.
And that plasma cell can create a large number of antibodies. I can't remember exactly what number, but I think it's like 10 to the fifth antibodies per day, or 10 to the sixth antibodies per day that is produced from that plasma cell.
Yeah, antibodies specific to the what did you say? The epitope of these?
That Yeah, that epitope or that region of that for that pathogen.
Yeah. Okay.
Yeah. And so if there's a new disease or new pathogen that comes in, it goes through that same process where a B cell has to activate to that disease. If now there are cases where that pathogen can be very similar to that old pathogen, and in that case there is a chance that your memory B cell could activate to that pathogen, and then it can start creating antibodies or forming that germinal center more quickly.
Yeah. Okay, cool.
Michael,
Yes.
very nice talk. I have several questions. So, um, first, you mentioned about the T-independent B cell and T-dependent B cell. So I'm curious how we can distinguish between these two, because as you mentioned, one of these, like T-independent one, is not creating memory cells. So then if we are doing, in terms of experimentally, I'm saying if we're doing experiment and it's not making the memory B cell, how we can be sure that it's going through that pathway, or it's not working or something like that.
Well, that's a good question, and I think that's what they're trying to find more markers to kind of be able to distinguish the two better. The hard part is that a lot of these B cells have the same markers, and so being able to distinguish them becomes difficult. But if I remember reading correctly, there are certain markers that you can look at to distinguish the two if you really want to see which pathway you're activating. But I think that if you inject what's it called? If you inject a large amount or a certain amount, or do different dosages for like a vaccination case, I think you can activate both pathways. I don't think you're limited to one pathway.
Mm-hmm.
So even though at first a T-independent B cell might activate first because it's easier to activate that pathway, when you have more antigens being introduced, I think you're giving yourself a higher chance that you'll also activate a T-dependent B cell activation. And even looking at, I think the paper you sent me with the Q beta, it
Mm-hmm.
mentions that Q beta will activate both pathways.
That's true. Yeah.
And so I think it just takes longer to get to your T-dependent pathway, which in literature it says can take up to 4 or 5 days for it to go through that pathway because it has to go through the germinal center. It has to
Mm-hmm.
find a helper T cell, and so that's why having a T-independent B cell is very helpful in the beginning of an infection.
Mm-hmm. Okay. Yeah, that makes sense. I have another question on slide 17. So you mentioned, could you
Hold
bring
on, yeah.
Actually, just I have a question about type of injection. So it was in the front paw. What kind of injection is it?
Okay, so for they did some subcutaneous injections, but to test the cell-mediated immunity, they do a paw pad injection.
What's
And
the
so
advantage of that, like
so
compared to other?
so it's more harmful to the mice, and not many people are doing this anymore. But what they do is basically they're avoiding looking at T cells, and what they do is they inject the paw, and when they inject the paw, you'll get a swelling effect, and they measure the swelling effect based on how much cell cellular toxic immune cells will go into there. And so the more immune cells you have that are going into that area, the more swelling you'll get. And so that's what they base the cell-mediated immunity response against. Does that make sense?
Yeah, yeah, okay. Yeah, it was interesting. I haven't seen something like that. And I think you mean my last question, I think you kind of mentioned it. So for you said regulatory B cell is secreting IL-10 or something like that. And I know that regulatory T cell is doing same thing. So
Yep.
is it difficult to distinguish between these, or like is there any specific way for regulatory B cell compared to that? Or like, I was kind of confused, like if we are measuring IL-10, so what it's saying that it's because of regulatory B cell or regulatory T cell or
So
something.
I think what they do is so you can still use CD45R, and then you can also use CD19, but then you would also want to add like CD1D or CD5, and that will tell you it's B regulatory. And then you
Mm-hmm.
can look at if you can then look at what it secretes.
Oh,
And
okay, that
and
makes
if you
sense.
look at interleukin 10, then you can be like, okay, you know that it's a B regulatory cell.
Okay, first
And
you
if you're
should prove
looking,
that this is that type of B
yeah,
cell, and then you go, okay, that makes sense.
yep. And then for helper T cell, you would look at CD4 and FOXP3 plus, and then you would know that that's a T regulatory.
Okay, I think I will ask you later about Th17. So I haven't heard about that before. I know there is a different type of Th1 too, but I didn't hear about Th17. So maybe later you can explain me.
Well, that's also kind of a newer T cell, and I would need to read more about it, but
Okay.
I do know a little bit, and they think that it might be involved in cancer response or tumor response. So,
Okay.
but
Okay.
they're still looking into that and trying to understand what it's for or why it gets activated.
Okay. Thank you so much.
I had to
Okay.
build off of that question. Sorry, to clarify, you were saying that you would do the CD staining and do flow and be like, oh, these are B regulatory T cells, and then do the IL by ELISA, IL-10.
No. Okay, so it's not B regulatory T cells. It's regulatory B cells or
Yeah,
regulatory
sorry.
T cells. And so
Yeah,
regulatory B cells, what you would do is you'd see the what it expresses on its membrane. And so you can look at CD19, CD45, and CD1D.
right.
And then you can also do a staining that's intracellular.
Right. Okay, for the
And
cytokine.
for the cytokine. And so
Okay.
then you could also do it by ELISA, but I think that'd be more difficult because it's hard
Right.
to separate the B cells from all. You can do it,
Yeah.
but it's very hard to separate the B cells from all the other cells.
Okay, that's what I was thinking. I just wanted to
Yeah.
clarify. Cool, thank you. And Jeremiah has a question in the chat, but he
Yeah.
says he
I
can't
was
say
gonna.
it.
Yeah, I'm gonna answer it right now. So I guess let me see. What is your interpretation of the final paper with 2R CF delivery method? Well, that I guess from that final paper that I presented, I just think we need to understand it more. I think that we're probably doing a too high of an injection. I see that we do get a cell-mediated response, but it's not that strong. And so I think that if we lower the dosages, we might be able to activate a stronger cell-mediated response. And I base this upon the fact that we're seeing a high amount of antibodies being produced. And that might be lowering our cell-mediated response. But I guess it really depends on what we're fighting and what pathogen it is. And so for bacteria, I don't know if it's better to have a cell-mediated response or a humoral response. And so if if you're looking at different pathogens, some people say that a cell-mediated response is better for that pathogen, or sometimes they'll say a humoral response is better. I guess that's something we would need to ask Molly, or I can look into it myself and see what we would prefer. And if you do want a cell-mediated response, we can lower our dosages and see if that improves it. But that's my thoughts on it. And I don't know if that answers the question fully, or if you have a follow-up question after that, go ahead and write it.
Well, I got my mouse charged a little bit. Well, so more broadly than that, I think that last paper, I sort of made an argument of that prolonged exposure to the same amount of antigen produces a stronger humoral response.
Yes.
So what we have, if we have a long-term release strategy, this is a strategy that's going to favor a humoral response, right?
Correct.
So it really doesn't necessarily, matter. It's just like this material is geared to initiate a stronger humoral response, which, I guess it does depend, because if you're looking at something like COVID, you want at least a strong neutralizing antibody response, I would guess. Or sepsis, I think the idea behind sepsis is similar, that you really are just trying to find and remove as much bacteria as you can. I don't know.
Right.
We'll definitely run it by Molly. I think this is sort of like where immunology butts up against, progression. I'm not sure what she'd say, but I'm pretty sure her contention in at least urinary tract infections is the production of antibodies to inhibit bacterial adhesion. So essentially neutralizing antibodies that block the cell surface markers that bind to the skin or the bladder or the epithelial tissue.
Well, I guess one thing, and I guess the reason why I'm not entirely sure is because when she presented her work, it seemed like she was getting more of a Th1 response, because she was showing that there was more IF and gamma and TNF alpha, and the activation of macrophages, and she even mentioned that she had a hard time seeing a high amount of antibodies, and so that's why I wonder if the body prefers a Th1 response for bacteria, but maybe, like you're saying, I would think that antibodies would be easier or better at clearing the bacteria, and so maybe it would be better if we could perform or develop a stronger humoral response against these bacteria. I would think it'd be better to have a humoral response versus a cell-mediated response. But I guess my only concern is that when she talked about the bacteria that lay dormant inside of cells, I wonder if a cell-mediated immunity could help prevent that from occurring, but I'm not entirely sure.
Right. Hmm. All right. Well, we'll run it by her when we talk to